Welcome back to session 7 of GED Science Mastery. Good to have you here. DNA is the instruction manual for building and running every living thing. The GED wants you to know what DNA is, how its letters pair up, and how genes connect to your traits. And here is a bonus. This session also sets up the next one on heredity, so getting it solid today pays off twice. Let's open the manual. Picture a giant cookbook stored inside the nucleus. The whole book is your DNA. One single recipe in that book is a gene. The book is divided into volumes, and each volume is a chromosome. Every recipe tells the cell how to build one protein, and the proteins are what actually do the work and shape your traits. Things like eye color or how fast a reaction runs. So the chain is the whole book is DNA, a volume is a chromosome, a recipe is a gene, and the finished dish is a protein. DNA has a famous shape, a double helix, which just means two strands twisted together like a twisted ladder. The two sides are held together by rungs, and each rung is made of a pair of chemical letters called bases. There are only 4 bases, and you should know them by their initials. A for adenine, T for thymine, C for cytosine, and G for guanine. Just 4 letters, A, T, C, and G, spell out every instruction in every living thing. The order of those letters is the code. Here is the single most important rule in this session, and it never changes. A always pairs with T, and C always pairs with G. That is it. A with T, C with G. Because the pairing is fixed, if you know the letters on one strand, you can always figure out the other. A handy memory hook is to think of the pairs as AT and GC. Whenever base pairing feels shaky, just whisper AT, CG. You will use this rule constantly. So how does a string of letters become a real trait? Step by step. A gene's sequence of bases is a code. The cell reads that code and uses it to build a specific protein, linking together small pieces called amino acids in the exact order the gene specifies. And proteins do the real work. They build structures, they speed up reactions, they carry things around. Those jobs add up to your traits. So the flow is the gene is the code, the protein is the worker, and the trait is the result. Let's get the packaging straight, because the GED tests it. A chromosome is a long DNA molecule wound up with proteins, and each chromosome holds many, many genes. So a gene is just a segment of a chromosome, never the whole thing. In humans, a typical body cell has 46 chromosomes, arranged as 23 pairs, with one of each pair coming from each parent. So remember, many genes sit on one chromosome, and 46 chromosomes make 23 pairs in you. Let's use the pairing rule on a real question. A short strand of DNA reads ATGCA. Find the matching strand. Read, the given strand is ATGCA, and the rule is A with T, C with G. Choose. Pair each base in order. A pairs to T, T pairs to A, G pairs to C, C pairs to G, and A pairs to T. So the matching strand is T, A, C, G, T. Check. Every A lines up with a T, and every C with a G. No forbidden pairings, so the answer holds. 2 more ideas the GED likes. First, replication. Before a cell divides, it copies all of its DNA so that each new cell receives a complete set of instructions. Both daughter cells need the full manual. Second, a mutation is simply a change in the DNA sequence. And here is the part people get wrong. A mutation is not automatically bad. It can be harmful, It can be neutral with no effect at all, or it can even be helpful. And those rare, helpful changes are the raw material that powers evolution, which is coming up soon. Let's lock in the traps. 1. Wrong pairing. It is never A with G or C with T. It is always A with T and C with G. 2. Thinking a gene equals a chromosome. A gene is only a segment. A chromosome holds many genes. 3. Thinking DNA does the work. No, DNA stores the code, but proteins do the work. 4. Thinking all mutations are bad. Many are neutral or helpful. The recap. DNA is the cookbook. A pairs with T and C with G, and genes code for the proteins that shape you. Excellent work today. You can now describe DNA's shape, apply the base pairing rule, write a complementary strand, and connect genes to proteins to traits. Your practice for this session is the 10 questions attached to this lesson. Answer them from memory, and if base pairing ever trips you up, just whisper A, T, C, G before each question. To read the full lesson notes and practice more GED science questions, visit the course page at www.bridgetoexam.com. I am really proud of you. I will see you in session 8, where we predict inheritance with Punnett squares. Keep going. You are doing great.